So welcome everybody to Structure 2, as in we love Structure 2. And it's my fantastic pleasure to introduce to you Cynthia Wolberger, our keynote speaker. Cynthia has been a leading figure in the field of structural biology. She has particularly shaped our understanding of how transcription and DNA repair are regulated by ubiquitin and chromatin, a sentence that has all my favorite subjects in it. So I look very much forward to this. Cynthia has studied physics at Cornell, then moved into biophysics, in particular is X-ray crystallography. The absolute vogue at the time, one has to say, as a PhD student with Steve Harrison at Harvard. There she solved one of the first structures of a DNA binding protein, which was really a huge deal at the time, as a first step towards understanding how proteins can read out to DNA sequence. And I know that for me personally and Brenda, this has been really key stuff at undergraduate teaching. So it's really shaped us and the feeling of our thinking about DNA binding proteins. Cynthia then moved to Johns Hopkins, where she ultimately set up her lab. There she took a deep dive into transcription factors, solving first structures of one of the largest subfamilies, the homeodomain transcription factors, and showing how transcription factors assemble on DNA in a combinatorial fashion. Well, this alone would have been plenty for a very successful career in science, but Cynthia just got going at that time, was just warming up. She then moved into more and more complex molecular machines that work on DNA, and of course, together with her colleague, the late Cecile Picard, started to work on ubiquitin. And after all, ubiquitin histones were the first proteins found ubiquitinated, and Cynthia focused on histone H2b in particular, and the many beautiful structures from her lab have very much told us how these machines kind of attach, recognize, and remove ubiquitin modification. From histones, she very much provided structures of the who is who in chromatin modifiers, such as FACT, COMPAS, DOT1L, sirtuinins, to only mention a few. And there are many, many more seminal contributions to the ubiquitin field as well. She provided the first structural explanation of how polyubiquitin chains are formed with her work on UBCH13 and complexes with E2s, and showing us how K63-linked polyubiquitin chains are made. And through her work on SAGA and O21, Cynthia further provided a deep understanding of how deubiquitinases work. And I have to say, from my point of view, watching this from the sidelines, I was always impressed by the elegance of her work and particularly the clever technical tricks her lab used to get these structures. I always ask myself, how did you do that? And the papers and the work has really been fantastic in that regard. So her honors are many, as you can deduce from that list of achievements, and include the membership to the Academy of Arts and Sciences, the Dorothy Hodgkin Award, and recently the ASCR Award for Outstanding Achievements in Chemistry and Cancer Research. And to those who have interacted with her, you will surely agree that she's a wonderful colleague with a great sense of humor. She's very passionate about teaching and mentoring, so dedicated that she actually wrote her own textbook. And Cynthia is also a great role model and a strong advocate for science and for women in science. And it's a great pleasure to have you here, Cynthia, and we look very much forward to your talk.
Oh, well, thank you. Thank you for that really amazing introduction. I really appreciate it. And really, I want to thank you, Nico and Anne and Micha, for putting this meeting together. I'm so sad we can't all be together. This is my favorite conference. This is the Ubiquitin community is a wonderful community and of people who share their ideas and their reagents. And this is the best meeting. So for all the younger people out there who haven't been there before, I encourage you to come back in 2 years if you can attend in person. It's really a fantastic meeting, and the talks have been amazing. So with that, I want to talk, and actually Nico gave you a bit of a preview of some of the things I'm going to talk about. I was messaging with Brenda. We were on a Zoom meeting last week together for the National Academy, and messaging. I was like, I don't know what to do for my keynote. I was thinking of talking about some stuff for the past, and she said, "Oh, do it. Talk about some retrospective." So I'm going to give you a little bit of history and then as well as current going all the way to unpublished work. So, okay, I will share my screen now. And okay, can you see, is the full screen visible, my slide?
Yes, just swap the display so we don't see your presenter tools.
Oh God, this is so weird. Last time it was the other way. Okay, all right. Okay, and no gray square, but I don't see the clock here. Is that going to become visible, the timer? Ah, okay, I have the, oh, sorry, I'm going to, I apologize.
Otherwise, the chair will stand up.
Yeah, I'm having a, well, I'd like to see the countdown clock, but oh well, I guess I won't. I have a clock here in my room. I will watch it. Okay. Okay. So anyway, all right. So I as Nico told you, I started my career working on transcription. So I actually thought I'd give you a little bit of a timeline as to how I ended up in the ubiquitin field. Oops. Okay. So as Nico told you in the beginning, I worked on the regulation of transcription. And really the interest, starting from when I was a graduate student, was in understanding how proteins that bind DNA and regulate transcription actually recognize their binding sequences and stimulate transcription. And when I established my own lab, we worked on combinatorial regulation. So we solved lots of structures of pairwise proteins to try to understand the interplay between them and how they regulated transcription. So that was about the first almost 10 years of my independent lab. We moved into studying sirtuins from the sirtu family, really because originally they were identified as being involved in transcriptional silencing, although that field blew up as it turned out that sirtuins had far more to do with many biological processes and not just targeting chromatin. So the way I got into the ubiquitin field was thanks to Cecile Pickart. And Cecile Pickart is really one of the founding members of the field who did seminal early work and who died all too tragically of cancer when she was just 51 years old. And Cecile's contributions are many, and I wanted to list some here. So she discovered, working as a postdoc in Ernie Rose's lab, that there were actually multiple E2 enzymes, that there isn't just one E2 enzyme. She did really beautiful work on dubs, including finding that, creating the ubiquitin aldehyde dub inhibitor. She was able to show that the E225K could actually synthesize free K48 chains. And really for a while in the field, she was the person who knew how to make K48 linked tetraubiquitin. Now we take it for granted that we can buy chains even, but at the time that biochemistry was difficult, and Cecile was a master biochemist and was master or mistress of doing that. She went on to study characterize the role of tetraubiquitin, collaborating, doing structural studies, showing how it recruited substrates to the proteasome. She did a lot of work on E2s, and as Nico mentioned, she discovered this enzyme, an E2 UEV heterodimer, that synthesizes K63-linked polyubiquitin as part of the DNA damage response. And it was through that we started working together, really through a completely chance encounter. So Cecile she started her career at the University of Buffalo and moved to Johns Hopkins around, I think it was 1998 or 1999. And by pure chance, she lived in the same apartment complex as my student, Andy Vandermark. Andy was a student in the lab who was trying to crystallize various protein DNA complexes. Nothing was working, the years were ticking by. And he came to me one day and he said, I was talking with my neighbor, Cecile, and I had met Cecile since she came to Hopkins. And, she just purified this heterodimer that makes K63-linked polyubiquitin. Could I try to crystallize that? And, you want to help a student who's in trouble and, be good to, collaborate with a colleague who wants to know some structure. And so Andy embarked on purifying UBC13 MMS2. So UBC13 is an E2 and MMS2 is a UEV. It's an E2-like protein, but it lacks the catalytic cysteine. And this heterodimer Cecile had shown catalyze synthesis of K63-linked polyubiquitin. So as they say, the rest is history. Andy got crystals. He saw this beautiful structure of UBC13 MMS2. We did some mutagenesis in collaboration with Cecile to come up with some ideas of how it might make K63 linked chains. And this was really my entree into the field. So as Nico said, we went on with Cecile's help to figure out how this heterodimer actually synthesizes K63 linked chains. And Cecile had the idea of making a stable E2 ubiquitin conjugate by mutating the active site cysteine to a serine and then making the oxyester. So with donor ubiquitin linked via an oxyester linkage to UBC13, which is much longer lived than a thioester linkage. And we were able to crystallize that, and through some fortuitous interactions in the crystal, we had a second ubiquitin that was bound here to MMS2, which immediately told us how linkage-specific polyubiquitin chains were made. So there's a K63 pointing to the active site. And so UBC13 catalyzed formation of this isopeptide linkage and charge the ubiquitin again, and so on and so forth. And you build up the K63 linked polyubiquitin chain. And this shows the linkage here. So Cecile, oh gosh, I have the wrong reference here. My apologies. This is Edens et al. So Cecile sadly passed away, but we continued our work on ubiquitin. And actually we began working on this Autobane-1, the K48 specific dub, while Cecile was still alive, and then continued that work in collaboration with Bob Cohen, who was at Hopkins in her lab and stayed on for a few more years, where we had a very fruitful collaboration. So working on this K48 specific dub, which then turned out amazingly to inhibit UBC13, and the mechanism by which that inhibition worked was figured out by Reuven Wiener, who you heard talking this morning. And I had to show this picture because this is a photograph of Reuven at the Ubiquitin meeting at Cold Spring Harbor 10 years ago. And he's sitting there in Grace Auditorium. So I feel very wistful. Hi, Reuven, but also I wish we were all there in that auditorium together. Anyway, we worked on other aspects of ubiquitination, dubs and E2s, but I wanted to figure out a way to sort of marry our interest in ubiquitin and transcription. And as Nico said, in fact, and I only learned this after working on ubiquitin for several years, the very first ubiquitinated substrate that was ever identified was histone H2a. It's ubiquitinated at lysine 119, but it had been identified as this very mysterious chromatin-associated protein. Chopping it up and sequencing peptides, it was found that it had similarity to both histone H2a and ubiquitin. And it was this paper in 1976, Goldknopf and Bush, that showed that H24 had 2 amino termini, but only 1 C terminus, and that it had the ubiquitin linked via an isopeptide linkage to that internal lysine. So although this very first ubiquitinated substrate discovered really had to do with transcription, the field went on to discover the role of ubiquitin and characterize it in proteasomal degradation. But in fact, ubiquitination of histones plays an important role in transcription regulation. So monoubiquitination of histone H2a lysine 119 is actually a mark of transcriptionally silent chromatin. And it's really quite abundant in heterochromatin. And histone H2b monoubiquitination is a mark of actively transcribed genes. And that's the one I'm going to talk about. So here's histone H2b shown here in red, and it's ubiquitinated towards the end of this, the C-terminal end of the protein. It's lysine 120 in humans, and the corresponding residue in yeast is lysine 123. It plays a role in transcription, although, and I won't talk about this other aspect, but it is also important for DNA replication and DNA repair and in regulating nucleosome dynamics. So how does histone H2b monoubiquitination regulate transcription? What is it actually doing? Well, it's doing a number of things. So first, it stimulates methylation of histone. So histone H3, lysine 4, monodi and trimethylation, and histone H3, lysine 79, sorry, this should be monodi and trimethylation, are other important post-translational modifications that are important in activating transcription and stimulating transcription elongation. One of the curious things about H2b monoubiquitination is that it's transient. It's rapidly turned over during transcription. It seems that it gets ubiquitinated and deubiquitinated frequently during transcription. And mutations in the aspects of the ubiquitination of H2b negatively impact nucleosome occupancy. So it is somehow required for maintaining sort of proper organization of DNA into nucleosomes. So if we look across the transcribed region here, so this is the nucleosome-free region and where RNA polymerase II starts, and this is sort of what a open beads on a string, that's what an actively transcribed gene looks like. The distribution of monoubiquitinated H2b sort of peaks near the transcription start, but really is found all across the gene body. And this ubiquitination mark is required for these two other marks. One is H3K4 trimethylation. So this peaks near the start site and is important for stimulating the SAGA coactivator complex and helps assemble the pre-initiation complex. And then histone H3 lysine 79 is more uniformly spread across the gene body. So although both of these marks are stimulated by ubiquitination of histone H2b, the enzymes that deposit the marks are very different. So histone H3 lysine 79 is located here in the globular core of the nucleosome, and it is methylated by this monomeric enzyme called DOT1 in yeast or DOT1-like, DOT1L in humans. Whereas H3K4, which is located in this flexible tail of histone H3, is ubiquitinated by compass, which is this multi-subunit complex. So that's the complex in yeast, and the human homologue is MLL1. And the catalytic subunit, set1, is from a completely different family, structurally different and actually mechanistically different from DOT1L. So how does H2b ubiquitination stimulate these two very different methyltransferases to deposit ubiquitin in two very different locations? So DOT1L, the human protein, is about 1,500 amino acids, but the business end is here at the amino terminus, this catalytic domain. And we've had structures of DOT1 from both yeast and humans for almost 20 years, but no one had been able to figure out how it binds ubiquitin or how ubiquitin stimulates it. This fragment alone can be stimulated by H2b ubiquitination, but it contained no known ubiquitin binding motifs. And mutagenesis to try to locate the ubiquitin binding site was not successful in identifying it. Another mystery about this methylation was that one couldn't figure out how this enzyme actually could even get to its substrate. So the lysine 79 in histone H3 lies pretty flat along the surface of the nucleosome. And if you took any of the DOT1 structures, you can't build a model where that side chain gets up into the active site. It just doesn't reach in there. So something's got to change here. So to try to understand how this substrate is accessed and how ubiquitin stimulates it, we determined structures of human DOT1L bound to ubiquitinated nucleosomes. And this is the work of a really terrific postdoc, Evan Warden, and parts of it, he worked on this in collaboration with another postdoc, Nick Hoffman. So here's the nucleosome shown over here. This is ubiquitin conjugated to lysine 120, actually via non-hydrolyzable linkage, and in green is DOT1L. So first of all, the ubiquitin binding motif was staring us in the face. This is the portion over here. I'll show you, zoom in. There's this, I think of it as sort of a hand at the back end of the enzyme, rigid, doesn't undergo conformational change, with a cluster of hydrophobic residues that bind to this I36 patch. David Commander was talking about the different hydrophobic patches on the surface of ubiquitin. Most things bind to I44, but this is interacting here with these two leucines and the I36 patch of ubiquitin. So, okay, that's how it binds ubiquitin. So what is ubiquitin doing? So we actually trapped two different states of this enzyme bound to a nucleosome. One was an active state, which we solved to structure of 3 angstrom resolution, and this other poised state at 3.9 angstroms resolution. And I can, the method's published. If people want to know how we trapped one versus the other, I can explain that another time. So we're looking at the active state, and I just want to show you the morph between the two. So we'll dissolve away the cryo-EM map. And now this is the poised state over here with the enzyme sort of far up off the surface of the nucleosome, and then there's the active state. So ubiquitin is really serving as a pivot. It's holding that tail end of the enzyme, and it's allowing it to switch between a poised state and then an active state where it's sitting down on the surface of the enzyme. One of the things that helps to stabilize it is this tail of histone H4. And I'll come back to that. So you think of it as the ubiquitin kind of holding one end. Now, when it's in the active state, that tail of histone H4, which is otherwise disordered, comes up and is very ordered and is binding actually in a cleft in .1L. And here's lysine 79. This is S-adenosyl methionine. So that is the methyl donor. And then on this end, we have the ubiquitin and actually another interaction with the acidic patch. This is actually as we heard earlier from the talk by Sam Willis from Rachel Clevitt's lab, there's an arginine there that's interacting with that nucleosome acidic patch, helping to anchor it there. So one of the mysteries, as I told you, was how that side chain gets into the .1L active site. And we were counting on some sort of conformational change in the enzyme itself, because the core of the nucleosome is this globular rock. But to our great astonishment, it's the nucleosome that undergoes a conformational change. So that side chain swings up by 90 degrees, and that NH3 group is moving 10 angstroms, and now it's within striking distance of the methyl group of H3K79. So this is going to attack the methyl group, and that methyl group will end up on the lysine here. And I'll just show you that conformational change again. So I want you to watch. Here's .1L in the poised state, and watch what happens to that lysine as it shifts to the active state. So it really comes right up there into the active site where it can be methylated. So this was actually, I know this is a ubiquitin meeting, but from the chromatin point of view, this was very exciting because, as I said, there's over 100 structures of the nucleosome. Histone H3 always looks the same. And so having it actually undergo a conformational change was pretty amazing. And also, this surely is not the only case. So it actually has some pretty interesting implications for how effector proteins might recognize H3K79 when it's methylated. So now this conformational change is very energetically unfavorable because we rip apart an alpha helix smack in the middle of histone H3. But that tail of histone H4 that I told you is holding it there, it's not just holding .1 in the active state, it's actually assisting in this conformational change. So take a look over here. We're going to be ripping apart that alpha helix in order for lysine 79 to come swing up right at you. Okay, so now we've broken all these hydrogen bonds, energetically not good. But the tail of histone H4 comes in, and that arginine residue is forming multiple electrostatic contacts with those carbonyl oxygens which have swung out. So this is helping to pay for that unfavorable energy and helping to stabilize this altered conformation of this side chain, sticking it up into the active site. So you can sort of think of ubiquitin sort of serving like, I don't know, the tail and the legs of this robin over here. So here's the robin in the poised state, and it's kind of looking around for a worm. And every now and then it dips down and it pulls up a worm from the ground, just as .1L is pulling that lysine up off the surface of the nucleosome. And then that change is being assisted by the tail of histone H4, in particular, this arginine residue, arginine 19. And sorry, I forgot to mention, but actually it had been known for a long time that that histone H4 tail was quite critical to .1 activity. And actually, if you mutate this, it kills activity. So this explains now the very important role that tail of histone H4 plays in .1L methylation of H3K79. So that's how .1L is stimulated by ubiquitin. And now what about H3K4? So as I told you, this is methylated by a completely different complex. It's a very big multi-protein complexes that has been characterized by Ali Shalati and others. And this is the yeast complex shown here, which I'm going to talk about from S. cerevisiae. And it is conserved in humans. So the MLL1 complex, which is the human H3K4 methyltransferase, has most of these subunits in common with the yeast complex. The minimal subcomplex that is required for this activity had been characterized and consists of the subunits outlined here. Some of them are fragments, and this subcomplex alone is active on nucleosomes and stimulated by ubiquitin. And structures had been determined, this is of the yeast complex shown over here by Yorgos Skiniotis. Another yeast also at 2018. And this is a very similar structure to the yeast complex. So we focused on the cerevisiae complex and took this and made complexes with ubiquitinated nucleosomes and determined the structure by cryo-EM. So this gives you the big overview here. So, and this was determined at 3.4 angstroms resolution. So here again is the nucleosome. Now the different subunits have been color-coded. So this is this big beast that's sort of spanning the entire nucleosome. And DOT1L wasn't even touching the DNA at all. It's just sitting in the core of the nucleosome and touching ubiquitin, whereas the compass is all over the place. It's touching different parts of the DNA. It's contacting ubiquitin. It's contacting the surface of the nucleosome. Here's ubiquitin over here. It's looking smooth like this because the ubiquitin here is actually not as well ordered as it was in DOT1L and not as well ordered as the other subunits here, but we still get a pretty good picture of it. So how does this compare to DOT1L besides the fact that it's much larger and touches the DNA? So unlike DOT1L, which is really rigidly binds to ubiquitin, compass changes conformation when it binds the nucleosome, and actually conformational changes are needed to contact the ubiquitin. It recognizes a larger surface of ubiquitin, both the I36 patch, which DOT1L recognizes, but also the I44 hydrophobic patch. And as I said, there are no rigid ubiquitin binding motifs, just residues that undergo conformational changes to recognize ubiquitin. So here is the apo complex. So again, this is the cerevisiae isolated complex. And what I'm going to show you is a morph between that structure and the structure that we see bound to a nucleosome. So I've superimposed the nucleosome here. Here's ubiquitin as it is in the full complex. And I'm going to show you a morph now from the apo complex to the nucleosome bound complex. So you could see that it flexes over the surface of the nucleosome and so comes down over it. And actually these subunits are forming contacts with the DNA. A critical difference between the apo complex and the one bound to the nucleosome is formation of this helix over here. So this is called the RXR helix, and it had been identified by Bob Rader's lab as a portion of the SET1 protein that is critical for activity on nucleosomes and is required in order for this complex to be activated by ubiquitination of histone H2b. I want to show you the conformational change here, and then I'm going to show you the context with ubiquitin. So we're looking at a close-up now at a part of the SET1 protein. You're going to see this helix unravel in the complex with ubiquitin, and that turns into a strand that then nucleates formation of that RXR helix that's shown over here. It's called RXR because it's got a number of important arginine residues. So what's the helix do? So one end of this has a hydrophobic patch, and that's interacting with the so-called I36 patch. So with these three hydrophobic residues that are clustered here on ubiquitin. And then the other face of the RXR helix is interacting with the acidic patch of the nucleosome. Again, the acidic patch. Here are these arginine residues in that helix that forms, and here's the cluster of acidic residues on the surface of the nucleosome. This is in histone H2a. So that helix is really mediating contacts between both the ubiquitin and the nucleosome. Now we see there's a difference. The apo complex, the helix isn't there at all, and it forms in this complex, but it's really actually work from Ning Zhang's lab. So Ning's lab solved the structure of the K-lactis complex bound to nucleosomes with and without ubiquitin. And they found that this helix, nucleation and the full formation of this helix depends on that ubiquitin there. So ubiquitin is really playing a critical role in helping to shift the balance of this structure towards formation of this helix, which in turn forms these important electrostatic contacts with the surface of the nucleosome. So this is one aspect of ubiquitin recognition, and I told you that there's a second part that recognizes the I44 patch, and this is with a separate subunit, SWD1, it's called. And I'm showing you the morph from the apo to the complex bound to the nucleosome. So these residues come together, form this hydrophobic patch, and that hydrophobic patch is what's interacting with the I44 surface of ubiquitin. So I'll turn it around over here. So here's I44. We're looking at it now 180 degrees around, and we're looking face on to ubiquitin, how this cluster of hydrophobic residues is interacting with that patch. So this is just the cartoon that summarizes that. So the isolated compass, this is how it recognizes ubiquitin. We have this disordered region here and these residues that need to come together in a cluster, and then these need to form the helix. And ubiquitin is nucleating formation of this helix, which is then making critical contacts with the surface of the nucleosome. So what about other things that monoubiquitinated H2b is doing during transcription? So there's really a lot going on when RNA polymerase is transcribing. So RNA polymerase is transcribing through a chromatin template. And so what that means is that these nucleosomes need to be disassembled for RNA polymerase to transcribe through, and then reassembled in the wake of RNA polymerase. And that process is assisted by FACT, which is a histone chaperone that helps in reassembling nucleosomes that have come apart in the wake of RNA polymerase. There's ubiquitination of the nucleosome, and so that's mediated by this E2-E3 pair. So Rad6 is the E2, BREAM1 is the E3 ligase. In humans, this is RNF 2040. So that's monoubiquitinating ubiquitin. We have these deubiquitinating enzymes that are removing ubiquitin. Remember, I told you this is dynamic. This is going on. Ubiquitination, deubiquitination happens repeatedly during transcription. And there are two H2b dubs that have been identified in yeast. One is UBP10 and one is UBP8. And they're both somehow functioning in transcription. Then there are these methyltransferases, I just told you about DOT1L and COMPASS, that are methylating these residues in a ubiquitin-dependent manner. And actually that ubiquitin is also helping FACT, that histone chaperone that's assisting in either disassembling and, more importantly, reassembling nucleosomes. And ubiquitin is somehow required for the recruitment of FACT. So there's a lot going on here. And a big challenge to understanding what it's all really doing, how is this stimulating transcription, is trying to figure out the order in which this happens. When does the nucleosome get ubiquitinated? When does it get deubiquitinated? When is the methylation happening? When does FACT come on and go off? So we've been studying the two dubs that remove the ubiquitin. And even though they are both involved in transcription and they both have similar effects on levels of H2b ubiquitination if you knock them out, they really seem to have very different roles. So the UBP8, one of them is actually part of a larger complex. It's part of the SAGA coactivator complex. And it is itself part of a 4-protein subcomplex. And all 4 of those proteins must be associated in order for this to be active. The other dub is UBP10, and this is showing you a very low resolution cryo-EM map. This is, by the way, a crystal structure determined by Mike Morgan in the lab. several years ago. So UBP10 is a monomeric enzyme. It has a catalytic domain that looks very similar to UBP8. We don't have a structure, but it's the USP class domain. And then this long acidic region, we're not sure what it does. And this has different effects on transcription. It regulates subtelomeric silencing. If you delete this, it affects a different set of genes than this, sort of rather puzzling. So why are they same specificity, but they're somehow doing different things? And they're both found at actively transcribed genes. So we learned really actually accidentally through just doing some biochemistry and preparation to try to crystallize things that whereas UBP8 has similar activity on intact nucleosomes and on H2A, H2b dimers. So remember, I was showing you how the nucleosome gets disassembled. And one way it gets disassembled is the H2A, H2b heterodimers are popped out. And we actually didn't know, is the ubiquitin still on there? You know, does it deubiquitinate it before, after, during this, when this happens? And UBP8 didn't seem to care, but UBP10 vastly preferred H2A, H2b dimers over the nucleosome. This was pretty interesting. So is UBP10 looking for something in the disassembly process? And so we looked at then the activity of UBP10 on its substrates in the presence of FACT, that histone chaperone that is recruited by H2b ubiquitination. And this is work done by Mel Nune in the lab, who also is working on that cryo-EM structure. What Mel found was that this low activity of UBP10 on nucleosomes can be boosted in the presence of FACT. And actually, but FACT has no effect on UBP10 on the heterodimers, and actually has no effect on UBP8 activity. UBP8 activity is the same with and without FACT. And if you look at, these are experiments done by Tim Formosa, if you look at levels of H2b ubiquitination in a FACT mutant, this is a subunit of FACT with a mutation that impairs function, what we see here is a decreased level of H2b ubiquitination as compared to the wild type. So the ratio goes up from 1 to about 1.9. Even more, you're enhancing ubiquitination even more than deletion of UBP10 itself. So FACT is somehow working together with UBP10 in ways that we're still trying to figure out. And from other experiments that I don't have time to show you, we think that UBP10 actually has a global role in nucleosome assembly and disassembly, whereas UBP8 is more focused on deubiquitinating H2b, maybe near the transcription start site. And we have a model that we're currently trying to test. So we know that H2b ubiquitin recruits FACT, which helps in nucleosome disassembly. And so we speculate that perhaps UBP10 removes ubiquitin from H2b as kind of a gating mechanism to tell FACT that it can either put the H2A, H2b back in an intact nucleosome, or perhaps to release FACT from a nucleosome. And we're trying to test this now using single molecule methods. So I know my time is running short. And so I added, really inspired by the first sessions in this meeting, all the talk about drug discovery, which was very exciting. I thought I would put in a few last slides here of some efforts that we're making to look at inhibitors of DUBs, perhaps to use as drugs or as tools to study the role of H2b deubiquitination and ubiquitination in cells. And the focus here is on USP22. So USP22 is the human homologue of UBP8. And this is the H2b DUB that's part of this bigger complex called the DUB module, which is part of the SAGA complex. So this removes monoubiquitinated H2b, it's lysine 120 in humans. We heard a talk, I was very excited to hear the talk by Sjoerd van Wyk earlier, a couple of days ago. I learned that USP22 is a positive regulator of necroptotic death. USP22 has been associated with poor outcomes in a variety of cancers. It's high levels of USP22 are associated with resistance to treatment, and it also stabilizes SIRT1, which deacetylates p53. So we teamed up with Hiro Suga, who's at the University of Tokyo, to identify peptide-like inhibitors of USP22. we had a beautiful hypothesis based on structural studies in mutants that I won't take you through because the hypothesis turns out not to hold up. But for that reason, we thought that we needed peptide-like inhibitors. And Hiro has this system of this incredibly huge combinatorial library, up to 10 to the 12th cyclic peptides that he synthesizes and synthesizes them, leaving the mRNA encoding the peptides covalently linked via puromycin to the peptide, and then has chemistry to cyclize those peptides. And then they reverse transcribe the mRNA. So you have this massive library with every peptide tagged with the mRNA that encodes it. And so we sent human dub module to Hiro, and they went through multiple rounds of screening to identify the peptides that bind most tightly to USP22. And they synthesized them and sent them to us. And indeed, they were really quite effective. So I'm just going to show you a few experiments here. So the peptides, this is the chemistry. This is all published by Hiro Suga's lab for the chemists out there who are interested. The peptides ranged in length from 11 to 16 amino acids in the cycle. And they were really all submicromolar inhibitors of USP22. So this is a Kamasi stain gel. We're looking at the activity on nucleosomes. This is ubiquitinated H2b. This is, look at the free ubiquitin band being released in DMSO. And this is we knew was a dud peptide. And all of these peptides here really are great inhibitors of inhibitors of USP22 deubiquitination, removal of ubiquitin from nucleosomes. And this just shows one of our IC50 curves on ubiquitin AMC. They're as good as around 20 nanomolar and IC50. So really quite effective inhibitors. And we've taken them all and treated HEC293 cells with them. And all of them work most of them work pretty well in cells. So these are ubiquitin levels, H2b ubiquitin levels. So these are Western blots now in untreated cells. And you can really see enhancement of ubiquitin. It's probably better to look at these bar graph here. This is a summary of 3 Western blots. This is the level of ubiquitin in untreated cells. And so you want to look for an increase in ubiquitin levels. And this is comparing it to a pan-dub inhibitor, PR619. So really quite effective. And we've tested it on a number of human dubs, and it's quite specific so far on the ones we've tested. So this is DMSO control. This is inhibition of USP22 dub module. And you can see on these USP7, CYLD, and USP30, it has no effect on these and just a very tiny effect on USP30. So to summarize, so I've told you a number of stories of what H2b ubiquitination does in transcription. I showed you how our insights into how it's recognized and how it activates the 2 very different methyltransferase complexes, and that one of them, interestingly, remodels its nucleosomal substrate. I told you a bit about what we've learned about the distinct functions and activity of the yeast H2b dubs, and our speculation that one of them is involved in together with H2b ubiquitination in gating nucleosome assembly and disassembly. And finally, I showed you some of our recent unpublished results on novel peptide inhibitors that we've identified. And really the last point I want to make is there's so much more to learn about the role of ubiquitin in transcription. There's so much we don't understand about the timing, about the impact of ubiquitin. I haven't even talked about H2A ubiquitination. And of course, there are all the other ubiquitination marks that we've heard about at this meeting of histones that play roles in DNA damage, and there's DNA replication as well. So I want to thank the people who did the work. I think I acknowledge lab members along the way whose work I highlighted, and including some work by past members of the lab who worked on the ubiquitin aspect of our projects. I've had wonderful collaborators, Hiro Suga, Ashraf Brick, who we've done a chemist who's made synthesized ubiquitin derivatives, Tim Formosa, Mike Matunas, Bob Cohen. Bob, I think you're out there. Hi, Dave Fishman, and finally Cecile Pickard, really a wonderful member of the community who first got me into this wonderful field. And then finally, I want to thank these sources of funding. And lastly, I'd like to thank you for your attention, and I'd be happy to answer questions.
Thank you very much, Cynthia. That was fantastic. Questions? Maybe I start with a question. you're looking at these structures and thinking about what ubiquitin does to chromatin. So these structures are mononucleosomes, which are in a bigger context. So tetranucleosomes, higher order chromatin, whatever exactly that may be. But there's a lot of knowledge out there of the cytic patch, which most of these structures, I think, engage in packaging chromatin. So what does ubiquitination of histones or .1L binding or binding of any of these structures do to higher order chromatin and regulation of these processes?
Yeah, so so what I've read, there are really conflicting results about what it does. I think you can find papers that say H2b favors compaction, favors chromatin opening. You know, it's easy since it's more in the middle of the nucleosome. So H2A is on the end. You can imagine how it sticks out of heterochromatin. H2B, you could imagine as a steric block. There's a recent paper from Alwyn Kohler saying that it has something to do with liquid-liquid phase separation. I take some of those reports with a little, well, I remain skeptical until we learn more about what that is really doing. You know, one thing we've talked about doing, but we haven't gotten around to is, you know, trying to use more biophysical methods to look, as you say, look at longer chromatin fibers, because we're looking at isolated nucleosomes. And as you know, because I know you've done very beautiful work on this too, we're working usually with that Whittam 601 sequence, which was selected to wrap tightly around a nucleosome and be crystallizable. And that, you know, most natural sequences don't, even nucleosome positioning sequences don't bind as tightly. So it would be very important to look at everything that you and I are studying and others really in longer chromatin templates. And, you know, now that we could do things with cryo-EM, it's still challenging to assemble those templates with all the right modifications in them, but that's obviously the thing to do.
Great. So there's a question from Thibaut Mayo. Beautiful structures and talk. Maybe I missed that. Does ubiquitin also promote direct or indirect the placement of H3K79 near .1L, near the .1L active site? Or does promote H4R19 action?
Oh, sorry. I think there were too many parts to that. So I so H3K79 removal, was that one part of it?
Yes.
So the only histone methyl mark for which there is no known demethylase is H3K79.
Okay.
I think there's got to be one out there, but no one has found one. So you know, when methylation was first discovered, it was thought that simply by recycling the histones was the only way you got rid of the methyl mark. Honestly, that doesn't make sense to me for activated transcription. Right. So there's got to be a demethylase. What was the other part of the question?
The second part was how the H4R19 relates to that. So what promotes the action of that particular residue to pop out the K79?
So I mean, it's just there. There's actually a second arginine residue. I didn't show you all the details. There actually are two arginine residues, in fact, that bind in there. And it's this nice deep pocket. And it's really this nice binding site for the tail of H4. So nothing that I know promotes it. Now, there's a recent paper from yeast.1 from KJ Armash showing that lysine 16, which is acetylated, actually regulates the binding of that tail to .1. So that's really very appealing that it's an additional layer of regulation where, you know, histone modifications can modulate at least the ability of that tail to stimulate, at least in the case of yeast, it's been shown.
Great. Maybe in the sake of time, one more question from Ning that I also wondered about. But basically, what's the magic that the cyclic peptides go into cells in these assays? What concentrations are used or what tricks are being used? Because they usually have the reputation of not going into cells. But
yeah, so there are no tricks. So we used 5 micromolar peptide and incubated it with cells, and we're pretty surprised that it worked. To be perfectly honest, there's nothing. I didn't show any sequences. I'm going to submit this and post the preprints. So if anyone wants to, there's nothing obvious.
Great. I think maybe the remainder of the questions we put in the Slack channel.
Okay.
And thank you very much, Cynthia. And I would like to hand it over now to